And why this can become so difficult is let's just take a look at a process of a simple wet granulation, compression, and coating for a tablet. And these are the different process factors that you need to evaluate to see which of these might be a critical parameter that you need to study. So we need to determine which of these are a critical process parameter and which of these might affect the critical quality attributes. Now, hopefully we don't have to study each of these, but we do need to make sure that we address each of these. And the responses that we would look at are typically such things as this. For the powder blend, we want to look at the content uniformity. For granulation, we want to look at the size and shape and distribution for the particles. We want to look at the density, both bulk and tap density. We want to look at the flow rate, moisture content, and again, content uniformity. And for tablets, we'll look at such things as weight and hardness and so forth. So the ideal experimental design is going to be a screening study first so that we can determine which of those process parameters might be a critical process parameter. And we'll look at something like a Plackett-Berman design where we can do anywhere from 8 to 12 experimental batches at an appropriate scale. We want to review historical data and help us if we can eliminate some of the variables. We want to study as many parameters as we can and that's the reason we would do a Plackett-Berman screening design. We want to identify, like I mentioned, what are the critical parameters. And then we also want to perform, before we start this study, we want to perform some trial runs at the extreme conditions so that we can make sure that we can manufacture and get data from each of the runs that are going to be in this Plackett-Berman design. And what I mean by that is that we want to make sure that if we're doing a wet granulation and we're varying the amount of water being added, that at some point we don't add too little water, that we don't granulate at all, or that at some point we add too much water to it and that we end up with just a ball of material that we can't do anything with. So we want to make sure that any time that we're doing this, that we can do the work for all the runs. Otherwise, if we lose one of the runs when we do the study, that will really throw off your statistics. And then once we've done the Plackett-Berman, we would really like to follow that up with a full factorial of some point with center points. And the reason we do this is we want to characterize the magnitude of the effect. We use the center points to estimate what our variability is in the process. And that will help us, again, get the best information out of it as we can. We want to try to establish if there's any interactions between any of the parameters. And then we want to get to the point that we can do a response surface. So that can really help us define our design space. This will give us scientific justification for the manufacturing parameters. And we will usually end up doing multiple studies. We will do blending study, and then we'll identify what is the best blend parameters and process. We will take that process and do the granulation study, and then once we determine that, we will take the best of both of those and then do the compression study. So we don't do a blend all the way through. We optimize each step along the way. And again, we have to take into account and make sure that we can perform the trial at each of the variables that's in there. So let's now talk about a case study for some bead coating, and just to show you real quickly what type of data you can do with the properly designed studies. So our process parameters that we looked at for this is we were looking at spray rate, atomization pressure, product temperature, humidity, and pan speed. Now the first thing we looked at was we looked at the particle size that we got, both small scale and full scale, to make sure that our particles are going to be roughly the same from beginning to end. We ran our study, and we were looking at 2 different time points for our dissolution. We were looking at 30 minutes and 60 minutes. And this is a statistical analysis of the different parameters that we looked at. We found one that was significant, interaction between, or excuse me, yeah, one was significant. The interaction between spray rate and temperature, and the interaction between humidity and temperature was not significant. In this particular table, any value that is greater than 5 was considered to be significant. Someone has asked a question, in my experience, what are the most typical issues and challenges that innovators face during scale-up? Probably, I think the biggest challenge that I see is when you start to have variability in your raw materials or your excipients and the API, when the API is constantly changing so that it's very difficult to build up a database. The second biggest challenge I see is lack of API, so it becomes very difficult to scale the material up, meaning there's no drug available to do a lot of work at a larger scale. Part of it depends on the process. If you're doing a direct blend and tableting, that's much easier to scale up than if you're doing a wet granulation. Part of that depends on issues would be, do you have the same equipment train going from beginning to end? Meaning, if you're doing a wet granulation on a small scale, does it operate the same as what you would see on a larger scale? And that's probably where I see more problems than anything else, is just trying to keep consistency of both your raw materials and equipment and having enough API available to be able to do the work appropriately. A lot of times you're asked to do things very quickly with not enough material. You've been working at a scale of, let's say, 500 grams, and then all of a sudden you're asked to jump up to 25 kilos or more. And depending on the process, that can be a very big jump, and it can be sometimes very challenging to come up and make sure you get the same material. So going back to our presentation here, what we can do with this data that we collect is, as I mentioned, we can put together these type of plots, and you can look at what is the effect of these different variables, pressure and temperature, on the dissolution amount that you get at 3 hours. And the other thing it shows you is that at certain conditions it may have a positive effect, and at certain conditions it may have a negative effect. So it's very important to look at this, because as you see in this particular case, if you look at your amount dissolved at 3 hours, it varies from about 2% all the way up to 40%. So you get a very wide swing in this particular amount. And in this particular case, if we look at the effects of pressure and temperature, what you can see is almost a saddle effect, where you're going from a high at 44, you then go down through a minimum, but then you end up back at almost the same amount again as well. So it's not as easy as this is going to be a linear plane that goes through, but you can also have saddle points as well that go through here. So you have to take all of these into consideration. And in this particular case, if we're looking at these conditions and looking at the effect of humidity and spray rate on pressure and temperature, excuse me, you see very minimal effect. You can vary your temperature from 24 up to 30, and you can vary your atomization pressure from 2 up to 4, and you're getting very, very minimal effect on your dissolution. It's basically going from 19% up to about 21%. So in some cases, your variables will have a large effect on your result, and in some cases, such as this, your variables are going to have very minimal result. So basically, we've tried to very briefly step through today what is your objectives during the scale-up process. it's very hard to define a particular scale or anything like that, because a lot of it's going to depend on what the project train is going to be. It's going to depend on how much API is available. It's going to depend on what your critical processing parameters that you identify are. But basically, I think the message that we want to take away from this today is that the more you can study this early on, the easier it is going to be to transfer it to a commercial facility and to manufacture it reproducibly commercially. But the key is that you want to be able to at least do some minimal work, design of experiments, or however you want to call it, to help you identify what is the design space that you can operate in to still reproducibly manufacture that product. And if you set this up appropriately and you follow the steps and go through doing the DOE type of work and doing that type of statistically designed process, then what you'll end up with is you'll have a product that is quality by design. And if you get to that point, then it's going to be much easier to get your PAI through with very, very minimal questions at all. So now at this point, I've concluded my presentation. If there are any questions that you would like to ask, I would be more than happy to try to answer them.